Hi, my name is Dario Vignali. I'm the Frank Dixon Chair of Cancer Immunology, full professor, vice chair of the Immunology department, and I also co-run the Cancer Immunology program at the Cancer Center. Here I'm introducing myself and my research. We have focused for over 30 years on studying inhibitory mechanisms in cancer and in autoimmunity. Our research spans really from discovery through basic research to translational research, using both mouse models and analyzing samples from patients. We, on the discovery side, use extensively all kinds of systems immunology approaches, particularly single-cell RNA-seq, TCR-seq, site-seq. We also have people in the lab that are fluent, so to speak, in R code for themselves, even generate algorithms. I have a full-time staff member who will help students to become familiar with these kind of computational tools for those that are interested. And we use that heavily on the discovery side. From a basic research perspective, on the mouse side, we make extensive use of a wide variety of mutant mouse models. Some we've generated ourselves. We've probably generated over 20 mutant mouse models. We also are starting to use CRISPR, as many groups are, both for analysis of single molecules, but also from a discovery perspective. And we've also started to get quite heavily into protein engineering, both from a basic research and a translational perspective. We also have some studies where we analyze structure function questions through collaborations with structural biologists, but in-house do a lot of super resolution microscopy and high-end techniques to study molecules on the cell surface and how they signal, particularly the TCR and inhibitory receptors. From a translational perspective, we make ample use of mouse models of cancer and autoimmunity to study new therapeutic approaches that we've developed in the lab, and to translate those into the human setting. On the human side, we again use extensively computational approaches using single-cell RNA-seq. We're expanding our capabilities around multispectral imaging, and we will often analyze not only samples from patients pre-treatment, but also obtain samples from clinical trials that we've initiated that are from patients receiving standard of care anti-PD-1 and novel immunotherapies. And indeed, much of our research has led to some of these therapies that are now in the clinic, particularly around LAG-3 and PD-1. From a logistics perspective, what underpins all of this effort is spans across two different labs. My main lab, so to speak, that focuses on all of the mouse work is right here in the Department of Immunology in the center of the university. I have a second lab in the Hillman Cancer Center that I co-run with a former trainee of mine, now tenure track assistant professor Tulia Bruno, and we co-mentor all the people in the lab that focuses on all these human studies. In terms of our impact, I'm a highly cited researcher. That's the top 1% of all research scientists in the world, have been for the last 6 years, but that's not really a reflection of me. It's actually a reflection of all the trainees that have been in the lab. We've published over 200 papers, over 30 have been in high impact journals, and many of these studies, as I mentioned, have led to therapies that are in the clinic. I have extensive mentoring experience. I've trained over 60 postdocs and grad students. Many of those grad students, in fact, all so far, knock on wood, have each had a first author paper in a high impact journal, that's Immunity, Nature, Immunology and above. And 15 of those have got extramural fellowships. F31s would be relevant for grad students. Lastly, I would be remiss if I didn't point out that Pittsburgh is really a fantastic city to live in. We as a lab, not at the moment because it's COVID, but pre-COVID and certainly post-COVID, do a lot of social activities together. We try to do something every couple of months. We have retreats that are off-site. And it's really a great city to live in. It's big enough to feel like it's a real city, but it doesn't have the congestion and high prices, which is of course very important for grad students. So we're delighted you're interested in this program, and I'm certainly willing to accept students that are interested in rotating in my lab. Thank you for listening.